Today I will present about recombinant DNA technology. Recombinant DNA technology is technique involved in the construction, studies and use of recombinant DNA molecules. It is a technology of gene cloning. Alright. Gene cloning. Gene cloning is involved technique to manipulate DNA to produce recombinant molecules that could multiply copies of the target gene.
Tools in gene cloning. Firstly, target DNA. Target DNA contains gene of interest from eukaryotic cell or donor cell. Next is DNA cloning vector. DNA molecules that carry gene of interest. The characteristics to be amplified. First, can accept foreign DNA in the multiple cloning sites (MCS). Can replicate freely in host cells because of the presence of replication initiation. Process selectable genetic marker. Example, marker of antibiotic resistance (AMPR). Next, restriction enzyme. The restriction enzyme is cut or cleave DNA at specific palindromic sequence. It is cut or break the phosphodiester bond. There are two restriction enzymes, which are EcoR1 and SMA1. The EcoR1 is cut at 5' GAATTC T', producing sticky ends. It is cut between G and A and produce two sticky ends. Next is SMA1. The SMA1 is cut at 5' CCCGGG T'. It produces blunt ends. It is cut between C and G and produce two blunt ends.
We can use modifying enzymes. DNA ligase joins the gene of interest and cloning vector to form recombinant DNA. Next, host cell, such as a bacteria or E. coli. Cell that receive a recombinant DNA for amplification purpose. Characteristics can receive a recombinant DNA via transformations. Second, can maintain the structure of recombinant DNA from generation to generation. Third, can apply the gene product from the recombinant DNA.
So these are the steps to gene cloning. The first step is isolation. So isolate plasmid from bacteria. Isolate target DNA, gene of interest from donor cell or eukaryotic cell. Second step is cut or cleave. Cut both plasmid and target DNA using same restriction enzyme to have complementary sticky ends for successful combination. The third step is insertion. Insert gene of interest into open plasmid. Join them using DNA ligase, forming recombinant DNA. The fourth step is transformation and amplification. Recombinant DNA is introduced into host cell through transformation. Allow the recombinant DNA to be amplified.
The last step for gene cloning is screening. Culture the bacteria in a medium containing ampicillin and X-gall. To identify the bacteria carrying recombinant DNA, the bacteria that carries recombinant DNA cannot hydrolyze the X-gall because LAC-Z is not functional and cannot synthesize B-galactosidase. The bacteria that doesn't have recombinant DNA will not survive the ampicillin as it does not have the ampicillin resistance.
And now we have a song for you.
Zegre.
Isolate plasmid from bacteria. Isolate target DNA from donor cell. Cut a plasmid from DNA. Oh, you need to use the restriction enzyme. Join them using DNA ligase. Forming recombinant DNA. to transformation, and then we do amplification. Recombinant DNA is introduced into the whole cell, they do transformation, and allow recombinant DNA to amplify, and then we do a process of screening. Bacteria does not contain recombinant DNA. Oh yeah, it's one survive in ampicillin, as it does have ampicillin resistance. Bacteria does not have a gene of interest.